enough funding from, I'll just start with that sentence again. So thanks to everybody for coming, and this is funded from a combination of grants from the National Science Foundation, the Simons Foundation, and various NIH agencies. Okay, so there we go. So first of all, hold on, I've got a little tiny laptop here, so I need to minimize the window with all the people so I can see what I'm looking at. There we go. Okay, so what are aptamers? Sorry, my computer keeps on foiling my attempts to look at my own slides. Try that again. Okay. Okay, so what are aptamers? Aptamers are single-stranded nucleotide polymers, basically single-stranded DNA or RNA. That's what we're working with, the DNA in this particular example. They're selected for high affinity binding to specific targets. So we're looking for targets that are relatively small in size. We're looking for aptamers that have reproducible chemical synthesis. They're biocompatible, have low immunogenicity and structural stability relative to proteins. And for these reasons, they're advantageous in biosensing and therapeutics. So if you actually find one that works, you're kind of in the game to do a lot of interesting things with it. So this is basically kind of a shot in the dark. You're looking at very large combinatorial libraries of possibilities. So you have 4 to the n sequences of possible aptamers you can consider, where n is the number of nucleotides in the random variable region. And we're going to be talking about a process called CellX, where you take many random combinations of these single-stranded DNA candidates, and then you run them through an iterative screening process to see which ones show up as possible candidates to have high affinity for your particular target of interest. Okay, so next slide. Okay, so now we can move on to CellX. So, okay. All right, so CellX is systematic evolution of ligands by exponential enrichment. This is a technique that dates back to the early 1990s. Basically, you start with a large combinatorial library with randomized sequences, and then the ends of your long DNA strand will be constant regions that are basically used for things like PCR amplification and identification, and they also may bind together to form kind of like a stem region. I'll show you on some pictures in a little bit, which is kind of similar to a portion of a double-stranded DNA that you would expect to twist up into a helical structure in 3D. So typically what you do with CellX is you have your targets immobilized to a stationary phase, and then this library of candidates is passed over the stationary phase. There's eluting and via competition and PCR amplification. And this is done in an iterative process until you get a much smaller number of candidates that are possibilities for binding to your target. And then you can use next generation sequencing to identify the specific sequences in the pool that you're left with. And CellX is successful. It's not successful all the time. You can go through this process that's rather laborious, and maybe 30% of the time you actually get ahead. So this is an area where machine learning can make this process much more successful. Okay, so here's what we've done. We've looked at the CellX process, which is shown on the left, and then we want to have a flow chart that takes us from the output of CellX, which is typically a list of 5,000 on the order of, right, 5,000 aptamer sequences, then we want to encode or be able to calculate very quickly the 2D structure of these aptamers. So basically, DNA is, I'll have another slide on this, but DNA is a single-stranded DNA is a backbone with a sequence of nucleotides, and there's only 4 of them, and each one of those 4 has a sort of a partner nucleotide that it likes to bind to. So think of this as like, like suppose you take a very long piece of tape off of a roll of tape. If you don't watch it, the tape tries to stick to itself, and that's basically what the aptamer likes to do as well. Only there's an order to how it kind of sticks to itself. And so already there's a challenge to figure out what the optimal secondary structure is. So that's kind of a known problem, but it's not known in a way that is useful for kind of high throughput, numerical calculations. So one of the first things we did was develop a code that could compute many of these things at once. Okay, so then you have your 2D secondary structures. Then what we'd like to do is use machine learning ideas to sort down and down select from these 5,000 aptamers to about 1% of them, maybe on the order of 50, where you can do bench experiments, because those take time. You're not going to do bench experiments on all 5,000. So we actually have more information than just the aptamer sequences. When we get to the 5,000 aptamers, we actually have count rates too. So each one of these sequences will have associated with it a count that comes from the amplification. And so the high count ones are going to be obvious candidates, but there may be other ones that we want to look at as well. And the question is, how do we identify those? So we're going to use machine learning ideas involving things like similarity, clustering, topic modeling, very, very well-known ideas from machine learning to try to sort through this large list of aptamers. So that's the idea, that's the process. Okay. All right, so here are some challenges on the computational side. So first of all, as I mentioned before, we need fast computation of thousands of aptamer secondary structures. Okay, so what's the current state of the art for that? So if you talk to a chemist, what they do is they use a CAN package. The one that is most often used and relied upon is called mFold. It's 20 years old. It's a proprietary algorithm. It has an internet GUI interface. So basically you go on the internet, you go to that interface and you hand input the entire sequence that you're interested in, and you wait a little bit and then it tells you a secondary structure. So that's obviously going to be very cumbersome. There's no source code available. And so we'd like to be able to take that process and understand what's going on under the hood and generate our own code to solve for the secondary structures. Okay, and then what we'd like to do after getting the secondary structures is we'd like to develop machine learning methods that can understand the relationships between these structures. So there is some work in that area, and the current state of the art does not use the global secondary structure. The existing methods use pieces of motifs as that are part of the primary sequence. So we'd like to move past the existing methods for that as well. Okay, so let's talk about how one computes DNA secondary structure. This is not a new problem. It goes back to the 1980s. But what we'd like to do is we'd like to cast it in a sort of modern lens, thinking about it as an inexact subgraph matching optimization problem, which gives us some hints about how to do this a little bit better than how it's currently done. So we can think of the DNA secondary structure as a bipartite graph matching problem. So the graph is the DNA strand, so it's just a linear graph. It's a sequence of nucleotides that are, so some sequence of nucleotides. There are 4 nucleotides for DNA: adenine, which is A; guanine, which is we refer to as G; cytosine, which we refer to as C; and thymine, which we call T. So you have the sequence of A's, G's, C's, and T's, and it's just a linear graph. So it's very simple. And the edges of this graph are the DNA backbone of the primary sequence. So now we need to look at the fact that the nucleotides have conjugate pairs. So the A likes to pair with T, and the C likes to pair with G. And these are by far the most common way to get secondary interactions. And we're going to view those pairings as our matching, as as the local match that is part of this optimization problem. So the goal is to discover the combination of secondary interactions between nucleotides that achieves the lowest free energy structure. So there's some physics in this, and it's non-trivial. So now I have to go through how that all works. Okay. But before we do that, I want to refer to some of the work that's gone on in subgraph matching. So this is an area of research unto itself. And there are some chemistry problems that have been studied with subgraph matching, just not this particular one. This is generally an NP-complete problem. Algorithms for subgraph matching go all the way back to Ullman's tree search with backtracking method from 1976. Similar methods to that one, include VF2, VF3, getting into the 200s and the 2010s. More recently, there's been work to develop constraint propagation methods. So my group has done some of this work in the last few years. My student Yurin Ge and Dominic Yang. There's also some groups in Europe that have state-of-the-art codes for subgraph matching, like the McCreesh paper and the Glasgow code. So we've used the Glasgow code from that group and Christine Solnin's group as well. So you might wonder, what about deep learning? Yes, there are works using deep learning. One of the most recent ones that I'm aware of is AEDNet, which was published in Pattern Recognition last year. But that paper itself admits that the kinds of graphs that it can work with are typically still smaller than what is achievable with the constraint propagation methods. So for larger graphs, the constraint propagation methods are really the best state of the art. And we're going to actually use some of the ideas from constraint propagation for our algorithms. Okay, so, and when I say constraint propagation, I'll show you with an example for the DNA. So here's my example. So this is a serotonin aptamer, and we're going to look at how to view this as a constraint propagation problem. So in step 1, we create a matrix, and the matrix has the entire sequence in both the rows and the columns on the left edge on the top. And you can see it's very small because it's a fairly long sequence, and I just can't blow it up so easily. But trust me that those little colors over here correspond to the different nucleotides in the sequence, and they're exactly the same up here on the top as they are going down here. So the first thing you do is you look at all the possible candidates. So you say, okay, what are all the possible pairings with the A pairing to T and C pairing to G? And I'm going to color all of those boxes in green. So this is my full candidate list for possible connections. Okay, so that's it right there. And so that's step 1. And then step 2 is our filter, and that's what we do with constraint propagation. So we find a way to smartly eliminate candidates to make the solution space that we have to search through much smaller. And so the elimination here that we're using is the following idea. If you look at the energetics that you need, so you have to get into the weeds now for the energy calculation, and you discover that it's much more favorable when you have a connection, say, between a C and a G right here, that you have another connection here. In this case, it's an A with a T right next to it, and together they form a 2-level stack. And so you can see this is an actual structure for a DNA aptamer, and you can see that there's this middle region here, which has like a ladder structure. It's sort of a long stack. And if you didn't have the other pieces up here and down here, this would be the kind of secondary structure that would lead to your double helix in 3D. It would just spiral. And in fact, when you look at 3D solvers for the 3D structure and you give it something like this as a secondary structure, the whole thing tries to twist up, including this stack. So that's something that we're not working on yet, but we've kind of done some preliminary, kind of quick look at how that works. So here's an example right here. So once you get to step 2, you're nowhere near done. This is just a first step to kind of cut down the number of candidates that you have to look at. So then step 3 is your expensive step, which is to minimize the free energy over all possible combinations of secondary structures. And the way we're going to do that is how it's been done for years in the chemistry literature using phase classes. Okay, so the next slide is on the phase classes. Oh, there we go. Okay, so there are 4 phase classes that are typically categorized. There's a hairpin loop like this, and notice there's a step.
I cannot see your slide.
You
Yeah,
can't.
we lost your slides.
Yeah.
Okay, let me try that again. Yeah, the Zoom gets a little weird, so let me try it again. Zoom. Oh, that's weird. I see you guys, but I don't see.
Yeah, but I don't see the slide.
Yeah, no, I know. I don't see how to share screen either. That's just bizarre. My Zoom is very weird. Okay. I must have hit some button that did something odd. Now I don't see the button. You know, there's like a space bar button where you can click on share screen, and that's just disappeared, but let me just see if it's behind something here.
Yeah, maybe it's hidden.
I just don't see it. How bizarre is that? Oh, wait a minute. What's that? No, that's not it.
Should be on the bottom of the Zoom. Right.
Yeah, no, it's just gone. It's just completely gone. Huh. I might have to like close Zoom and come back.
Yeah. Okay.
I think I'm gonna do that. I'm sorry. That's just a mess, but what else can you do?
Yeah, no problem. Yeah.
I'm gonna hold on. Let me just see if there's anything else here. I don't see how to fix it other than killing Zoom and coming back. So let me do that.
Okay.
Okay. Okay, I'm back.
Then I will make you the co-host.
Yeah, except that I still don't see that bar. Oh God. That's just weird. Yeah, it still disappeared. That's just really weird. Okay, I wonder if there's a view, something under view. No. Full screen. No, share screen. Huh.
All right, share screen.
I'm just trying to find it. You know, there's like a horizontal bar, right, that has that on it. The horizontal bar is gone. And it's still not there. Like I tried to click on the next slide and it wasn't scrolling the slide. So I clicked on it again and then everything kind of self-destructed.
Andrea, it's not at the bottom of your screen?
No, I have the horizontal bar for the Mac, but not for Zoom. Oh crap. Let's see, view, edit, help. I'll just go to help and see. No, that pops up like a whole web interface. That's not helpful. Yeah, it's like every once in a while it changes, you know, it updates, and this may be a faulty update. I don't know.
See. Yeah, I don't know. I'm not. There's someone sent a message. Yeah, I can. You can send me your slide, and I can show on my screen, and you
Yeah,
can
I
ask
think
me
that's the
to.
right thing to do. Let me give you the
Yeah.
link to the
I
slides.
see. Mhm.
Let me send you the link to
And
the slides. Fortunately, I have them on Dropbox, so
Mhm.
that should be straightforward.
I have your slides from your last talk, Andrea.
Do you have the final version?
I don't, I think so, but I'm not 100%
You want to
sure.
try sharing screen? We could do that if you have them.
Yeah.
Yep, sure.
Okay, let me allow you to share it.
Okay.
Thank you,
Come
Anne.
on.
That's great.
Mm-hmm.
I have like zombie
See if
Zoom
this,
or something here.
okay. Let's see if
Yeah,
this
make you
is-
the co-host so you can try to share screen.
Okay, thank you.
No problem.
Um,
Yeah.
Can you see my screen?
Yes.
Awesome.
Okay,
Nice to have collaborators.
Hmm.
Yeah, we're on this slide, the face classes.
Okay.
Okay, beautiful. Okay, beautiful. Okay, so here are the face classes: hairpin loops, inner loops, bulges, and multi-branches. And the multi-branches are the most challenging to calculate because of the combinatorial complexity. Okay, so next slide. Okay, so we have a recursive process for computing the minimum free energy, and this is basically done in all, you know, all the literature uses a recursive process. So going back to the original paper by Zucker and Stiegler in 1981. So basically, one stores content in structure classes SV and SW. These are functions from a portion of the sequence between values I and j. So the indices denote a position along the primary sequence. And so these caches correspond to both an energy and a structure, which is, you know, basically some combination of phase classes in different positions along the sequence. So SV is the optimal energy and structure of the substring ij, assuming I and j interact, and SW is the optimal energy and structure of the substring with no assumptions. And then you basically you know, have to iterate on this process where you have a simple minimization problem to calculate SWIJ from you know, from a smaller set of sequence structures. There's just a simple minimization. Okay, let's go to the next slide. Okay, so one thing that we've done with our code is to improve the efficiency of the caches. So we avoid unnecessary recursive passages by computing the caches in a specific order. E.g., we compute the SW for very small subsequences with just like k equals 4, and then increase them outward. And this was not how it was done in earlier codes. And by following this idea, no unnecessary recursive passages are needed, leading to a reduction in the computational cost of order n, where n is the total number in the of the total number of sequences in the part that we need to calculate the optimal free energy. Okay, let's go to the next slide. Okay, so now let's talk about codes. So in terms of open source codes, there's a code from last year called SecFold. It's the first DNA specific open source Python code, and it was never published in a journal. So it's kind of an alpha version of a Python code. So one of the things we did was we took that code. When I say we, I mean Maddie and Paolo did a lot of work on this. So so we call our improved version SecFold 2. And what SecFold 2 does that SecFold doesn't do is it uses this cache efficiency idea and it uses the graph matching approach in steps 1 and step 2 from the previous slide. And also we found that there were a number of incorrect face energies in the original secfold code, so those are improved. Okay, so let's go on to the next slide. Okay, so secfold 2, by making these changes, and in particular the efficiency changes, give us a speed up on the order of a factor of 2 for secfold 2 versus secfold. So these are just some runtime calculations on the same computer, just benching one code against the other code. For sequences with various lengths. And I should add that we're typically not looking at lengths of 400. We're looking at sequences that are in the 50 to 150 range. So we're down in the smaller end for at least the calculations that we've been doing recently, although there are plenty of examples in the literature where people are interested in longer sequences for aptamers. Okay, so let's go on to the next slide. The other thing we did was we wrote our own code from scratch called gmfold. So this is an and the goal here was to have an open source Python alternative to mfold, which is the current state of the art. So we use the energy function provided by Zucker and Stiegler, which is what mfold is supposed to do. And we use the step 1 and step 2 from subgraph matching, this constraint propagation part of the algorithm. So our multi-branch optimization is more robust than Zucker and Stiegler because we compute all combinations of at most edges in the substring. We eliminate the need for secondary cache, meaning we only need to store structure and energy information of the pair IJ if IJ is an admissible bond, reducing the size. I shouldn't say bond. So Anne likes to make sure I say something other than bond for these things, right, Anne? So, but anyway, so reducing the secondary interactions is what I mean to say.
That sounds great, interaction.
Yeah, okay. Reducing the size of the cache. So our code allows for different temperatures, which is important because you want to be able to understand what the structures are as a function of the temperature because they're not the same. And then there are some things that we would want to do in an updated version of the code that we didn't have time to do here, which is to look at different salt concentrations because that affects the structure. We set ours up for the most commonly used salt concentration. We have limited coaxial stacking stabilization energies. There's more complex ones in a paper by Santa Lucia and Hicks from 2004. And another key component is that all the admissible energies that we consider have to satisfy that step 2 in our constraint propagation. That means that we do not allow for pairing between non-conjugate nucleotides. And for the lion's share of the aptamers you're interested in, that's okay, but every once in a while you find one that mfold thinks should have a non-conjugate pairing. And so we have to get more into the weeds to figure out why those happen and how to adapt the code to do that. So this is where we are with our gmfold. So let's go to the next slide. Yeah, and I guess one thing I want to, for the group of people who are attending the talk from the AI side of things, one takeaway you should have here is this is a combinatorial optimization problem. This is, I mean, you're looking at a very large list of possible candidates that you want to consider for the optimization. So it's a little different from some of the more classical AI machine learning problems you might encounter in research, but it's a really important one in computational chemistry. So here are some examples on 5 single-stranded DNA sequences so secfold has a GitHub repository, so we picked 5 from that GitHub repository, and you can see the minimum free energy in the boxes on this table, and we're actually using, so you have to pick some code, like you have to pick a benchmark to calculate them. Once you get the secondary structure, what you'd like to do is use one benchmark for computing all the free energy. So we use mfold for that. So you can see that that the first 3 examples have gmfold and mfold getting the same lower free energy than secfold or secfold 2. And then for S4, secfold 2 and GM-fold do better than secfold, but M-fold has an even lower free energy. It's a slight change in the structure. This is a bit of an art, I should say. Like, this is not an easy problem. And even if you believe that some list of energies is the optimal, it still may not correspond exactly to what happens in the chemistry. So there's a bit of an art going on here, but we have to make some decisions as to what we're going to try to optimize. So this is what we're doing. And then S5 is an example where secfold does the worst, secfold 2 does better, GM-fold does much better, and then M-fold has an even lower energy. So these are just some examples. Let's go to the next slide. Okay, so S1 and S2 from table 1, so now we get to look at pretty pictures. So these are the aptamer secondary structures. You can draw them in the plane. And so what we're looking at is a single-stranded DNA that's folded along itself, and you can see that the folding results in a fairly complex structure. In this example, you see a couple of hairpin loops, and you see a multi-branch structure in the middle. And you can see that central multi-branch structure for S1 is bigger for secfold and secfold 2 and smaller for GM-fold and M-fold. So these are just, the way the algorithms optimize. And then for S2, you have the same kind of results where secfold and secfold 2 do not do as well as GM-fold and M-fold, which get identical answers. Okay, let's go on to the next slide. This is another example where secfold and secfold 2 get completely different structures. And so that has probably something to do with the incorrect face energies in secfold. But when we go to GM-folds, we get an even different structure. So you can see on that big kind of loopy thing at the bottom, there's an extra kind of small hairpin structure that comes off of it. And then for M-fold, it's similar, but it's not quite the same hairpin structure. So these are, and you can see also at the very top where you have the multi-branches that they're a little bit different between GM-fold and M-fold. Okay, let's go to the next slide. Okay, this is a biotin. Now we get some real aptamers. This is a biotin aptamer. And again, GM-fold and M-fold are not exactly the same, but they're very close. And secfold is somewhat different. And secfold 2 is kind of in between. All right, so let's go look at the next slide. Theophylline. Now this is an example where GM-fold, our current GM-fold code, where it doesn't get the non-canonical pairings. And so for some reason, M-fold has decided that a non-canonical pairing is admissible. And so we see that on the right-hand structure. So you get an extra piece of a stack that shows up in the middle of that folding that doesn't show up in either GM-fold or secfold. Okay, let's go to the next slide. Okay, so basically what you should take away from this is that interpreting these secondary structures is still a bit of an art, and that we spent a long time, and when I say we, I mean Maddie and Paolo mainly, with help from the chemists, spent months going through the literature, going back to the Zucker paper and so forth, trying to really untangle and unpack what these DNA folding codes are actually doing, to map it out. And I think there's still parts of it that we kind of view as more art than being exact. So I think we still have a little bit more to unpack, but I think we've done a pretty good job up to this point, to the point where we want to actually use GM-fold for machine learning. So here's what we did. So we have some recent data, thankfully, from Anne's group, and this is looking at target binding for norepinephrine, which is a neurotransmitter. So Anne is an expert on neurotransmitters. Serotonin is her primary area of expertise, but she knows lots of things about many of these different molecules. And so we have some data from a norepinephrine Select screen. And this screen uses lab protocols that are already published in the literature. I believe, Anne, that Casey Yang did some or all of this chemistry analysis. She can correct me here. Okay, she's quiet. All right, and there were two different libraries used, one with a 48 nucleotide randomized region and the other one with a 58 nucleotide randomized region. So these are fairly small aptamers. The library design preferentially selects for aptamers with a closed sum structure upon target binding and tertiary structure formation. So that's a component that's relevant to biosensing applications, which is an area where Anne has a tremendous amount of expertise and some really important papers in the recent literature. So let's go to the next slide. Okay, so here's what we did. So the Select screen is done. And then what we have from that is raw data that is about over 16,000 single-stranded DNA. So we start with like billions and we end up with 16,000 single-stranded DNA sequences and their associated counts from the amplification. So it turns out that this raw data includes duplicates. So we remove the duplicates and keep the sequence associated with the highest count, and we keep that count. Then we remove all the sequences with less than 20 nucleotides and those with an invalid nucleotide character. I'm not sure how those show up, but at any rate, that's what we do. Then we remove outlier sequences based on length, keeping only those with lengths between the 5th and the 95th percentile. So this is basically the data cleaning part of the process. And then we end with 4,450 sequences with lengths varying from 33 nucleotides to 83 nucleotides. Okay, let's go to the next slide. Okay, so what we'd like to do is we'd like to take this group of over 4,000 aptamers that now have very similar structures between themselves, and we'd like to sort them. We'd like to put them in clusters, ideally, but we have to figure out how are we going to use this new information of the DNA secondary structure to do that. So we start looking at ways to encode the secondary structure. So there are two ways that we've considered. One is the structural matrix of which nucleotides pair with which nucleotides, which you can write actually as a one-dimensional function called a Motzkin path, which is something that's very well studied in the combinatorics literature. So a Motzkin path, you can think of it as a discrete function that starts at 0 and that can increase by one step or decrease by one step, and then at the end you have to end up back at 0. So that's an example of the Motzkin path for this particular structure. So that's one thing that we can use to try to do similarities between DNA secondary structures. The other thing we can do is we can view this as a language, right? So we know that DNA is often referred to as a language for genetics. And so there's been a lot of work done over the last 20 years working with human language, with natural language processing methods. And so the simplest way to study natural language processing is using a bag of words kind of construction where you have a document. In this case, our document is one strand of DNA, and we've got 4,000 of these documents. And then what we'd like to do is we'd like to say, okay, what are the words in our document? Well, the natural words are the face classes, right? We already know this from the chemistry. So we can look at the number count, the histogram of the different types of face classes and their corresponding energy, and kind of come up with what we call a bag of faces approach. Which is modeled on the classic bag of words approach. So these are two different things that we can do. And then let's go to the next slide. Okay, so for the Motzkin path idea, what we can do is we can look at distances between individual DNA sequences, and we can do that by taking the Motzkin path, and you can write that as a sequence of zeros, ones, and minus ones. And then you can look at the Euclidean distance between different Motzkin path. You can view that as like a feature vector, right, in machine learning, and you can look at the Euclidean distance between these things, and then you can do a 2D representation of that whole set of aptamers using a t-SNE embedding. So this is t-distributed stochastic neighbor embedding, and you get a kind of a pretty picture. So here's the pretty picture. So every dot in the picture corresponds to one sequence. And we also have the counts from the Select screen for each sequence. And so you can see the high count dots are colored in yellow, and the low counts are colored in dark blue. And then there's a continuum of count values. So there are 4 sequences from the Select screen that have high counts. And so we call those x tilde 1 through x tilde 4, and you can see them in the picture here, the high count ones. And so the hypothesis that Anne and Noel decided to make was that maybe aptamers that have a low count, but a very, very similar secondary structure to the high count ones, maybe those could be of interest. So we can identify those very quickly by looking at these, from the t-SNE embedding, looking at the clusters and looking at the aptamers that are very close in this reduced dimension space to the high count. So what I'm showing you here on this picture is that for every high count aptamer, we have another one that's right next to it that's a low count, but it has almost exactly the same structure. It's not identical, but it's very close. So we were able to identify those very quickly. And these are now candidates that are undergoing additional testing in the lab by Anne and Noel in her lab. So I can't tell you the outcome of that yet, but that's where we are with this. Let's go to the next slide. So yeah, and right, and so I mean, we can get into the math on this. We can look at energy similarity neighborhoods and topology similarity neighborhoods between these different aptamers. So there's a way to actually write down some mathematical formulas for the neighborhoods that we want to look at for these aptamers using the Motzkin path structure and using the energy distribution. So let's go to the next slide. Yeah, so right, so we can use the bag of faces to compute energies of the sequences with the same topology as the high count aptamers, and we can take all of the ones that are similar to, for example, the X1 aptamer, right? And so that's shown in the left-hand graph. That's a histogram. And so what you see there are the bag of face descriptors, which allows us to divide all of those similar sequences. And what we see is that the group that has the high count aptamer has a total of 80 other aptamers that kind of match or at least are in the same bin as using bag of face descriptors. So this is just a way of kind of sorting through everything. We can do this in a very systematic way now with all of this information. We can do the same thing with X2, with X3, and then with X4. So let's go to the next slide. So here's another, so we can also start with a bag of faces and cluster directly based on that. So this is using topic modeling. So once we have our bag of faces, we have histograms of the face, each of these faces. We have a histogram of their use in each of the aptamers. So that's documents by words, if you will, or documents by faces. So that's a big matrix. So then we can take that matrix and we can do non-negative matrix factorization to get a list of topics. And then we can take the topics and we can do similarity graph clustering doing spectral clustering with those. So these are all, so we're basically using I could say sort of now classical methods from machine learning. When I say classical, I mean going back like 15, 20 years classical rather than like 50 years. But these are well-known methods from machine learning. And again, you find that you can sort these into clusters based on the topic modeling, and then we can visualize those nicely in 2D, again, using the t-SNE visualization. So this is just another way of grouping these things. Okay, let's go to the next slide. Okay, so the takeaway here from the topic modeling analysis is even if the clusters are computed from high-dimensional descriptors, the 2D embedding aggregates in specific regions of the plane. So this suggests that the similarity of the high-dimensional descriptors is well represented in our lower dimensional space. Also, the geometric distribution of the two-dimensional representation points in the same structure may aggregate in more than one region in Euclidean space. So, I mean, so there's some, there's some interesting kind of hidden structures going on here. The dimensionality reduction highlights additional hidden patterns between the sequences within the same cluster, which can potentially be exploited to segment the larger clusters further. So, I mean, I personally like this approach. I'm a big fan of these somewhat classical machine learning methods because they allow us to, like, there's no black box here. It allows us to completely unpack and sort through the entire population of aptamers that survive the Select screening process. And I think that's important for chemists if they want to be able to identify aptamers that might be possible candidates for target binding. Okay, let's go to the next slide. Right, so this is where we are. So this is where we are with the research. It's very new research where our paper is, like it's in the final stages of submission, so it's very new. So the conclusions are that we've developed a new Python tool for secondary structure of DNA aptamers. It allows for high throughput calculation of the secondary structure of thousands of aptamers. Using data from a Select screen, we show how to use machine learning methods to interpret the entire list of aptamers and their counts. We can identify low count aptamers that are really close in structure to the high count aptamers. These would, and this is an important point, these low count aptamers that are really close in structure would not have been easily identified with conventional methods. And further analysis of these aptamers is needed. So you're not done when you get to this stage. Like you have to go back and do the bench experiments for affinity and selectivity for the target binding. So that's another step in the process. And in summary, you can see that to do this, you really need to have very close interaction and collaboration between the machine learning folks and the chemists. Like you can't split this work up. There's got to be a lot of going back and forth between experts in both areas of the science to be able to do this. But I think it's a worthwhile problem to work on. I've been really I feel very privileged to be able to have worked with our chemistry colleagues on this problem. So I'm going to stop there. Thank you. Oh yeah, no, this gets into just extra slides. So I'll stop there. Thanks,
Okay.
Anne, for sticking me on the slides. I don't know what went on with my Zoom.
Okay, thank you. And I have a question that in your previous slide, you said that you have a sequence folding and a GM folding. What's the improvement from that you reach a smaller free energy from sequence folding to GM fold? What's the improvement inside these two algorithms? Is it that you have a better for the combinatory problem?
That's a good question.
Yeah, you have
right.
better
So
constraints.
if we think, right. So if we compare with M-fold, in theory, we have a more efficient algorithm, but we don't have the source code. No one has the source code except for the authors of M-fold to be able to bench. It's proprietary. So
I
we
see.
can't bench against theirs. But we think that so you want to do machine learning, you want to be able to develop a code that you can open source and put it out there, right? So that's what we've done. Now, whether it's better or worse, I mean, we think it's better than SecFold. We have a lot, or SecFold 2 even. We have a lot of evidence that it's probably better than SecFold 2. But we don't even know, frankly. I mean, according to my chemistry colleagues, whether even mfold is the ground truth. And what is the ground truth, right? This is a little bit of an art. And you should comment on this since you're here.
Yeah, I mean, that's exactly it, Andrea. We don't really have the ground truth. These are just predictions. We can do some experiments to, very laboriously determine which bases are most likely pairing with what, which other bases. And we have some other biophysical experiments that we can do to tease out some of the structure. But these small biomolecules, and because they have a lot of negative charge, they're really not amenable. They don't like to make crystals, so we don't have a lot of ground truth.
I see.
Yeah, it's a problem space where, I mean, and this is my understanding from chemistry in general, you don't always know, like you have a theory for what you're doing, but chemists, remember the state of the art way to do science and chemistry and biology as well is a hypothesis based analysis, right? So you have to make a hypothesis and then design an experiment to test it. They're not doing things from first principles like we like to do in math and physics. It's a very different way of doing science. You're trying to probe what's going on from the top down rather than from the bottom up. And so it's interesting for us to work with the chemists because we have to start to think like them, and we have to say, okay, so we've come up with a model for these sequences, but we know they may not be ground truth, but nevertheless, our model at least is going to be consistent across all of the sequences that we try to fold, right, in any particular list of candidates, say, that come out of a SELECT screen, and then we can try to look for similar structures in those particular sequences.
Thank you.
Yeah, these are really good questions.
Yeah, and I think also we have a longer range goal, which is, now that Andrea and her team have the algorithms to predict the secondary structures, I think the next bigger goal, which is a harder one, is to go after predictions of tertiary structure, which will be really exciting, and then to start to include the targets, in those tertiary structures.
Yeah, so there are canned packages for the 3D structures, but again, you have to input the thing by hand, and then they'll give you a visualization of it. It takes a while to compute. Like it's not set up to do what we're doing here. So we will want to write our own code for 3D and then try to figure out how to incorporate target binding. So that gets into so by the way, that's a very different modeling from what we're doing here, right? So this secondary structure problem is really a combinatorial optimization problem. It has it really has that kind of underlying math to it. When you go to the 3D structure, you're basically done with the secondary structure, and now you're looking at,
3D folding,
yeah, you're looking at continuum, like folding and twisting of that's how they do it, right? It's more of a molecular, like there are different ways you can do it, but one example that we all know very well from applied math would be a force law model, to take that structure and then look at individual types of force laws and how does it, typically there's some kind of twisting that happens. So yeah, and then once you have the 3D structure, how do you unpack that? How do you then sort through similarities in that space, right? So there's a lot of things to do. But yeah, even what we've done up till now is quite state of the art for the literature. Like nobody's done anything like this. in the existing Aptamer literature. It's a natural thing to do coming from my perspective, but there aren't that many chemists who are trained to do machine learning. So it was a fun thing to do, to get together and pair
Yeah,
up. I also
very
want to
interesting.
I want to give a shout out to the California Nanosystems Institute, which was very much responsible for kind of getting the chemists and the mathematicians to talk to each other, and even providing collaboration space for us to do that work at UCLA. So, you know, for people who are interested in trying to get into this kind of problem area, it really helps if you have a place where you can hang out with the chemists. Like, you don't want to hang out in their lab, trust me, there's got to be chemicals there. Chemicals, right? So yeah. So no, it's been a lot of fun. And thanks for letting me giving me the opportunity to talk about this
Yeah,
work.
it's a very nice talk. Yeah.
Thank you.
So any question from the audience? Okay. If no, yeah, thank you for your speaking today.
Yeah, you're welcome. Thanks a lot.
Yeah. Okay.
Okay, bye.
See you.